Well, good morning, and hello, everyone. I'm Sally Head-Delquist, President of Immunochemistry Technologies, and thanks for joining our webinar today. We're talking about lysosomes, and lysosomes are these amazing little organelles in your cells that do so much. They're more than just a garbage can. They also help repair the plasma membrane, regulate calcium, transfer molecules between other organelles, and even impact the immune system. And I'm really excited to tell you about some breaking news on the coronavirus, how it actually spreads using lysosomes from just last week. A little about me. I'm Sally Delquist, and I'm a biochemist. I have a BS in genetics and cell biology and an MBA. I started out working in the lab, developing assays for apoptosis, and now I'm president of the company. Immunochemistry Technologies is a biotech company, and we're on a mission to help cure disease, and we do that, ironically, by studying cell death. So our customers want to know if the disease or the treatment is killing the cells or keeping them alive. And one aspect of that is to look at organelles like lysosomes. If the lysosomes don't work, the cells will die. And we have some great reagents to study lysosome function, cathepsin activity, and how that relates to other processes like cell viability, apoptosis, autophagy, mitosis, calcium transfer, mitochondrial functionality, and toxicity. We have over 2,000 publications using our products and a lot of new publications from 2020. Assisting me today is Allison Westcott, our customer service manager, and Nick Weninger, our scientific associate, who helped compile a lot of this data. So a big thanks to Nick and Allison for helping. And to those of you out there watching live today or watching the recording in the future, we really appreciate you taking this time to make this webinar a part of your day. So we encourage you to ask questions. Just type them into the chat box or email them to help@immunochemistry.com, and we'll respond directly to you after the webinar. So take some notes or make a screenshot of the slide you like. And we are recording this, and we'll post it on our website and YouTube and email a link to you, so you'll be able to watch it again anytime, slow it down to look at your favorite slide, and share it with your colleagues. So we hope this data will give you some inspiration when setting up your next experiments to help you understand the unique biochemistry of lysosomes that will ultimately lead to discover better drugs. And that's why we're all here, to help cure disease and save lives, like COVID-19. So let's start with that. I'm just going to jump in with a technical paper. This was published last week. They found that coronavirus escapes through lysosomes. So scientists had assumed that coronavirus escapes through the biosynthetic secretory pathway, like the flu, hepatitis C, and West Nile, which you see on the top half of the picture. But now they found that it escapes through the lysosomes. That's the bottom half of the picture. So when they blocked the biosynthetic pathway, the coronavirus still escaped. And they discovered that coronavirus actually accumulates inside lysosomes, but it doesn't get destroyed by the acidic pH. Instead, the coronavirus takes over the lysosome and de-acidifies it. So the degradation enzymes that would normally destroy the virus won't work at a higher pH. The virus remains intact and infectious. And disrupting the lysosome also disrupts antigen presentation and immune response, which may explain why the cytokine storm is seen in some COVID patients, which overwhelms the body with pro-inflammatory proteins that can be fatal. Let's take a look at some of their pictures. This is transmission electron microscopy, TME, picture of an infected lysosome in the egress stage. It has dense material and membrane swirls and LAMP1, LAMP1 on the inner membrane. It is filled with intact viruses typical of 70 to 90 nanometer particles. Those are the blue spots. In a normal cell, there are often hundreds of lysosomes, and they can vary in size and shape depending on environmental cues and cellular needs like starvation. Healthy lysosomes are at a low acidic pH that is maintained by constantly pumping hydrogen ions, those are H+, by a vacuolar type A ATPase. That's the VATPase, which I'll talk about a lot more. But somehow coronavirus stops the acidification and turns off the lysosomal degradation enzymes. And there are 60 of them in a normal lysosome. So understanding how SARS-CoV-2 affects the lysosomal pathway will really provide vital insight to help stop the spread of COVID-19. It also highlights to me why lysosomes are so interesting, because they play a role in so many aspects of cellular health, often in unexpected ways. So lysosomes are an active dynamic regulatory hub that is intimately linked to multiple pathways that control cell and body homeostasis. Here is a really nice graphic published in 2020 by Andrea Bellabio from Naples, Italy, that I'll reference a lot. So thanks to them for all their research. So lysosomes are these amazing little organelles found throughout the cytosol of mammalian cells. They are membrane-surrounded organelles that arise from the fusion of the Golgi-derived vesicles with endosomes and are the primary catabolic compartment. So their main job is thought to be garbage disposal, breakdown and recycling of unneeded proteins and waste. But now we know that they do so much more. They also transport ions like calcium to other organelles, and they play a pivotal role in gene regulation and mitosis, cell signaling, repair of the plasma membrane, defense against pathogens, cholesterol metabolism, cell death, energy metabolism, and homeostasis. To do so many jobs, lysosomes also contain transport factors like NPC2, which shuttles free cholesterol for export out of the lysosome. They also have solute carriers that export sugars, nucleosides, amino acid, and snare proteins that mediate fusions of lysosomes with other organelles. We'll start by looking at lysosomes and their role in degradation. Lysosomes are filled with many hydrolytic proteases like cathepsins, amylases, lipases, and nucleases that enable it to break down all sorts of biological polymers, including proteins, lipids, carbohydrates, and nucleic acids. They require a low pH, so the enzymes don't work in the cytosol because it's at pH 7. The enzymes reach the lysosomes through the secretory pathway. They are synthesized in the rough endoplasmic reticulum, it's the ER, and modified in the Golgi and tagged with an M6P marker. The lysosomal associated membrane proteins type 1 and 2, those are the LAMP, L-A-M-P, they are highly glycosylated on the inner membrane to prevent self-digestion and make the membrane more stable. There's also LIMP, LIMP proteins, which are lysosomal integral membrane proteins 1 and 2, that stabilize the membrane and help with some M6P independent transport. So of all these enzymes, cathepsins in particular are often used as a biomarker for lysosome function. Let's look at them next. Cathepsins are a group of protease enzymes typically found in the lysosomes. There are 11 different human cysteine cathepsins. The majority are ubiquitously expressed in human tissues. For an example of structure, here is cathepsin L. It has 2 domains, right and left, and the reactive site is in yellow, in the middle. Cathepsins demonstrate broad specificity, meaning that they cleave their substrates preferentially after basic or hydrophobic residues, consistent with their roles in intracellular protein degradation. This decreased functionality is associated with neurodegeneration, like Parkinson's disease, while increased cathepsin activity is associated with many disease states such as cancer, especially if it's released into the extracellular matrix. Cathepsins are optimally active at a slightly acidic pH. In fact, they're mostly unstable at a neutral pH. And when outside of lysosomes or extracellularly, they can be rapidly and irreversibly inactivated. Cellular stress or triggering TNF receptors could initiate cathepsin release from the lysosomes, where they're translocated, and then they could go on to cleave cytosolic substrates like BID, which would trigger the release of mitochondrial factors, ultimately resulting in apoptotic cell death. Cells use a variety of methods to regulate cysteine cathepsins to prevent this type of uncontrolled proteolytic activity, which can be very harmful. First, the cell can compartmentalize the cathepsins within the lysosomes or other organelles. Second, the activation of the zymogen, or the inactive precursor to the active cathepsin enzyme, is tightly regulated. This activation occurs during the passage of the zymogen through the endoplasmic reticulum, providing a signal peptide so that the enzyme can be properly folded and sent to the lysosomes. pH is another factor in regulation. Cathepsins need an acidic environment to maintain structural stability. And finally, there are small molecule inhibitors such as cystatins, thyropins, and serpins that can bind competitively and tightly to the cathepsin active site to prevent proteolytic activity. Cathepsins have been used to study many specific diseases and also are considered an overall biomarker for lysosomal function. So if the lysosomes are healthy, cathepsins are active, and this activity can easily be detected and measured using ICT's magic red kits. For example, can lysosome function provide a clue for stopping the coronavirus? In this study, researchers are looking at the drug Artesunate, or ART. This is a malaria drug that has anti-cancer properties. They found that ART treatment activates lysosomal function via cathepsin L activity by promoting assembly of the VATPase. That's the hydrogen pump for lysosomes, which lowers the pH. ART then promotes ferritin degradation, subsequently leading to the increase of lysosomal iron that is utilized by ART for its cytotoxic effect on cancer cells. So if the lysosomal iron is chelated, ART cannot kill the cells. These are HeLa cells treated with ART that show enhanced lysosomal staining in the bottom compared to the control on the top. They were co-labeled with lysotracker green, which labels acidic organelles. And using these labels together means the lysosome has a low pH and functional cathepsin L enzymes. They did many other experiments and quantitated cathepsin L activity using the flow cytometry on the top. This makes me wonder if a drug like ART would work to stimulate the VATPase to lower the pH in lysosomes and make their enzymes functional again to destroy the coronavirus. On the other hand, VATPase can be inactivated by bifidobacterium A. So does the coronavirus act like bifidobacterium? Oh, and remember, we are recording this, and we'll send you a link and post it on YouTube. So you'll be able to look at it again in more detail, take your notes, make a screenshot, and certainly feel free to send us any questions to help@immunochemistry.com, even if you're watching this recording later. This particular study was on drug toxicity, so now let's look at how lysosomes might be affected by a gene mutation. In this experiment, they were studying a gene mutation in LYST, LYST, the lysosomal trafficking regulator protein. This causes neurological dysfunction, bleeding, and recurrent infections. The researchers wanted to create a cellular model of this disease and replicate the condition, and they successfully did that using U2OS osteosarcoma cells with a silencing and siRNA knockdown of LYST. They replicated the phenotype of fewer yet larger lysosomes that you see are bright red in the bottom picture with active cathepsin B compared to the top normal phenotype. They also stained the nucleus blue. They did many other experiments and found that most processes of the lysosomes are normal, except that there is an increase in mTOR localization to the lysosomes, and there are more lysosomes localized to the paranuclear area. So we'll backtrack a little bit and next talk about how does something get into the lysosome for disposal. So how does something get into the lysosome for digestion? What happens when something enters the cells? Items enter typically from the plasma membrane or the biosynthetic pathway. It's either degraded on the left and goes to the lysosome, or it's recycled on the right. And this is orchestrated by the retromer complex. Substrates for degradation enter the lysosome by fusing with phagosomes or via autophagy, either micro, macro, or chaperone-mediated autophagy. And items to be recycled would be, for example, a cell surface receptor and its ligand from the plasma membrane. And once that receptor ligand complex enters the endosome membrane vesicles, it dissociates in the acidic lumen of the endosome, and the receptor exits the endosome along with the bulk flow of membrane lymph nodes that are returned to the plasma membrane. These pathways can be corrupted in many different ways. So let's take a look at some lysosomal storage disorders. In neurons, lysosome degradation is critical to maintain cellular homeostasis. Nearly all lysosomal storage disorders result in neurodegeneration. And neurons are particularly sensitive to changes in lysosomal degradation because they are postmitotic, so they don't divide anymore, and the undegraded garbage proteins inside the lysosome simply build up over time as they are not diluted by cell division. This accumulation becomes neurotoxic and leads to cell death, leading to a progressive loss of neurons causing disease. So the majority of Parkinson's-associated genes are linked to lysosomal protein trafficking or lysosomal function. So one example is alpha-synuclein that aggregates and becomes toxic. It should be degraded by cathepsin B in the lysosome, but it isn't. So next, we'll look at if the lysosome is functional in the neuron. This is a neat experiment using transgenic mutant neurons that are a model for Alzheimer's disease. These mutant HAPP neurons have defective lysosomes, and our magic red reagent for cathepsin B was used to monitor lysosomal functionality. So cathepsin B decreased in lysosomes in the mutants on the bottom compared to the wild type on the top, and this is quantified on the graph. So mutant HAPP neurons have impaired lysosomes. You can clearly see the bright red spots in these pictures. In this experiment, they used GFP with magic red. In the next experiment, they used a highly pH-sensitive version of SEP to monitor the whole neuron. So in this experiment, they were looking at CBLN1, which is involved with synapse formation. They showed that CBEL1 is released from axons but not in dendrites, and is co-released with cathepsin B upon electrical stimulation and co-localizes in cerebellar granule cell axons. So this is a new mechanism of activity-dependent coordinated synapse modification. So next, we'll see that neurodegenerative diseases may also exhibit mitochondrial dysfunction, and how does that impact the lysosome? Mitochondrial dysfunction disrupts energy metabolism in the cell, which leads to the production of reactive oxygen species, ROS. It also disrupts the structure and function of lysosomes. So here they used a knockout mouse that was neuron-specific for AIF, that's the AKO mouse, which shows similar mitochondrial fragmentation and loss of complex 1 expression, which is a new model of mitochondrial-dependent neurodegeneration. This inhibition of mitochondrial function impaired lysosomal activity and caused large lysosomal vacuoles and is dependent on ROS, which you can see the large red vacuole on the lower right. Next, let's look at the mitochondria directly. These are human kidney 293 cells. In this study, they were looking at a single amino acid gene mutation that causes amyloidosis, which is the buildup of amyloid fibrils in tissues and organs. The fibrils target lysosomes that become overloaded. It increases lysosomal pH and causes the release of cathepsin B into the cytosol, which leads to mitochondrial dysfunction. Here they measured mitochondrial depolarization with TMRE, which is red in healthy mitochondria on the left, but TMRE loses its fluorescence when depolarized, as seen on the right. So a lot of these experiments use multiple labels, and before we look at data, let's take a look at some of these kits and stains real quick. So you'll notice throughout these experiments that they use many different stains at the same time to label cells in red, green, blue, orange, and many other colors. Some of them are even arbitrary depending on the computer program. And you'll see a lot of examples using blue nuclear stains, Hooks and DAPI, and others using the red vital stain, PI, the green fluorescent proteins, label the oligomers, and many more. Our kits are compatible with these, and many of them are used concurrently, like our Magic Red kit for cathepsin B is frequently used with our FAM Silica caspase 1 kit to measure the activation of the NLRP3 inflammasome. So you can correlate lysosome function with an immune response, particularly for toxicity studies. So let's look at two toxicity studies, one on inhaled silica particles and the other on metal oxide nanoparticles. So silica inhalation leads to inflammation and silicosis, which there is no cure and treatment is really limited. So particles like silica permeabilize lysosomes, which release their enzymes like cathepsin B into the cytoplasm, which then triggers assembly of the NLRP3 inflammasome, which activates caspase 1, which in turn activates IL-1 beta and IL-18 pro-inflammatory cytokines. But without the receptor MARCO, this response is exaggerated. So MARCO is a scavenger receptor that binds to toxic silica particles in lung alveolar macrophages, the AL. The key to silica-induced inflammation appears to be cholesterol recycling by MARCO. In knockouts without that MARCO receptor, there are two defects. Number one, less silica is being taken up, and number two, cells can't sequester the cholesterol, both of which increase lysosomal membrane permeabilization, and this triggers a greater inflammatory response. They were able to decrease the inflammatory response by increasing cholesterol using U18666A. They looked at cathepsin B activity for lysosomal functionality, that's the top bars, and caspase 1 activation in the bottom bars to measure NLRP3 in mouse cells. Cells were analyzed on a fluorescent plate reader to quantitate activity. So in the next experiments, they wanted to know if this type of hyperinflammatory response can be predicted. And yes, it can. So this is a toxicity study looking at metal oxide nanoparticles, which are used in paints, they're drug carriers, in food additives, and in cosmetics, and some of them can be hazardous. And we don't know how hazardous. And can we predict the inflammatory potential of these nanoparticles? Well, yes, you can. So like the last slide, inflammatory potential was based on IL-1 beta release, which is pro-inflammatory and is triggered by the NLRP3 inflammasome. They used HeLa cells and also examined lysosomal damage by imaging red cathepsin B co-labeled with hooks. And using their QSAR modeling, they can predict toxicity with 90% accuracy. So now we'll be able to predict inflammation. So going back to how lysosomes originate, this is another great review article published in May 2020 that I'm going to use quite a bit today. So thanks to the lab of Purvi Trivedi in Halifax, Canada. So lysosomes are membrane-surrounded organelles that arise from the fusion of Golgi-derived vesicles with endosomes and are their primary catabolic compartment. These endocytic vesicles, EV, from the plasma membrane mature into late endosomes and to lysosomes. And so lysosome biogenesis is also regulated during the cell cycle. Let's take a look at how lysosomes can be transferred to daughter cells during mitosis. Lysosome biogenesis is regulated during the cell cycle by the action of CDK4 and CDK6 cyclin-dependent kinases, whether they're turned off or on, which impacts the function of TFEB, which is critical because TFEB is a master regulator of autophagic flux by controlling both cargo delivery and substrate degradation. So what's happening in the nucleus here is that at G1, D-type cyclins, those are orange in the lower left, activate CDK4 and 6, those are the yellow circles, and they phosphorylate TFEB and TFE3 in blue, which are lysosome and autophagy transcription factors that would normally increase the number of lysosomes. But when TFEB and TFE3 are phosphorylated, that's blue with little orange circles, when they're phosphorylated by CDK4 and 6, they are exported into the cytoplasm, which inactivates them, which decreases the number of lysosomes. During S and G2M phases of the cell cycle, cyclin D turnover inactivates those CDK4 and 6, so TFEB and TFE3 are not phosphorylated, and they stay active in the nucleus where they promote lysosome biogenesis and autophagy, which increases the number of lysosomes you see on the right. Now we can actually see these functional lysosomes in their microscope slides, and they quantitated this lysosome function. Here they analyze cathepsin B activity using our magic red substrate assay in HeLa cells and duly stain them with hooks. They treated the experimental cells with an inhibitor, which is LY2835219, to turn off CDK4 and 6, which is the picture on the right, which should increase the number of lysosomes. And we see that on the right, they increase the number of red lysosomes in the cell compared to the DMSO control on the left. Then they quantified the red fluorescent signal from 30 cells and made the bar graph above. So both normal and experimental conditions show bright red lysosomal staining. However, the experimental condition on the right shows many more lysosomes, and the overall red signal was much brighter. In fact, it's 1.5 times the control, which you see on the graph. So you can compare the black bar control with the orange bar for the experimental side. These findings help explain the molecular events directing the nuclear export of TFEB and TFE3, and also suggest a mechanism that controls lysosome biogenesis in the cell cycle. Because it's so important, let's dig a little deeper into TFEB, which controls cargo delivery and degradation. So TFEB is the transcriptional regulator of many genes involved in the lysosomal autophagic pathway, shown here in the gray boxes. This impacts formation of the isolation membrane, conjugation of ubiquitin-like molecules of the ATG8 family, for example, LC3, elongation of the autophagosomal membrane, cargo recruitment, autophagosome-lysosome fusion, and lysosome-mediated degradation and lysosomal biogenesis. In the previous slide, they looked at TFE3 as well, which is another member of the same family, the MIT-TFE family, which also includes MITF and TFEC. So TFE3 is partially redundant for TFEB, and they coordinate together, and they have roles regulating the circadian rhythm, which links them to nutrient-driven gene expression. So there is an MIT-TFE and mTOR-C1 feedback loop, how lysosome function adapts to environmental cues. Under normal conditions, TFEB is mostly in the cytoplasm. It translocates into the nucleus upon starvation and other environmental cues. On the left in B, this Also includes bacterial infections, pro-inflammatory agents, physical exercise, endoplasmic reticulum stress, oxidative stress, and mitochondrial damage, which allow adaptation of the lysosomal autophagic pathway to cellular needs. The main mechanism that drives that is phosphorylation of specific serine residues. On the right, in C, we see how phosphorylation modulates nucleocytoplasmic shuttling of TFEB. For example, activation of the TRPML1 calcium channel in the middle results in TFEB dephosphorylation and nuclear translocation back into the nucleus, therefore turning it on. When TFB is phosphorylated on serine S142 and S138 residues, which is the box on the right, which these sites are dependent on mTOR, the mechanistic target of rapamycin, that promotes its binding to the exportin, CRM1, and subsequent nuclear export and turns it off. So in the cytoplasm, TFEB can be phosphorylated at S211, mediated by the mTOR complex 1 that induces binding to other proteins. There is an mTOR C1 and TFEB feedback that helps adapt to metabolic conditions. Overall, this relationship between MIT and TFE factors and mTOR C1 links global control of lysosome biogenesis to lysosomal nutrient sensing and explains how lysosome function can adapt to environmental cues. Next, we'll look at some dividing mitotic cells. So these researchers in Barcelona, Spain, were studying chromosomal instability, which is the abnormal gain or loss of chromosomes during cell division and correlates with poor cancer prognosis. They characterized an atypical phenotype that can now be used as a novel biomarker for genotox screenings. That's the toroidal nucleus, which looks like a donut on the left. The nucleus has a hole devoid of chromatin, but contains cytosolic material, including autophagic vesicles and lysosomes. The toroidal nucleus was generated as a defect in mitotic progression formed after karyokinesis. Depletion of motor proteins involved in lysosomal trafficking and movement, such as KIF3A, KIF2A, or KIF1A, also led to a significant increase in this toroidal nuclei. Also, it increased when acidification was disrupted. We found that depletion of the ATG5 inhibits autophagy, which significantly increases misaligned chromosomes and chromosome bridges. So there are two processes required to preserve mitotic integrity. Number one, that's this VATPase acidification, and number two is the BORC-associated movement, which I'll show you more about in other slides. So let's look at these dividing cells. I think these pictures are so cool. So mitosis relies on lysosome-dependent degradation, as impairment of lysosomes increases the mitotic timing and defects. So normally, lysosomes must function to degrade these mitotic factors that would be used to segregate the chromosome. But if these mitotic factors aren't degraded and instead they keep working, it increases mitotic timing, leading to mitotic errors, which promote chromosomal instability. So these images show that lysosomes are active in red when co-localized with lysosensor in green. Blue is for DNA, and these green and blue colors are arbitrary in this picture. So now we know a little bit about how cells divide. Let's take a look at how they fuse. An important role of lysosomes is to transfer cargo and ions, so they have to fuse with other organelles. Distinct sets of snare molecules drive this fusion. Additional factors such as small GTPases, tethering factors, autophagosome docking proteins, regulators, and calcium fluxes all contribute to the specificity, efficiency, and regulation of this fusion. So let's look at more at the Hops tethering complex, which is in the lower left. It's shown in the yellow box in C and D on the bottom. In this experiment, explores how lysosomes are tethered and fused with other intracellular compartments so they can transport macromolecules. They're studying the Hops complex, which is homotypic fusion and vacuole protein sorting. And that's required for lysosomes to fuse with endosomes, agosomes, and autophagosomes, the biogenesis of lysosome-related organelles, and possibly with signal regulation via AKT2, among other roles. So on another note, HOPS is also required for Ebola virus to enter the cells and in resistance of melanoma cells to cytotoxic drugs. Here they compared mammalian versus yeast because they have different subunits. So these are HeLa M cells and silence the gene activity using an siRNA to knock down any one of the 6 subunits. They wanted to see if the endosomal, lysosomal compartments were still functional. So they labeled them with magic red for cathepsin B to show they're enzymatically active. Then they wanted to know if the endosomes could fuse and deliver dextran to the functional lysosomes. So they added green labeled dextran with Alexa Fluor 488 to see if it would be taken up by the lysosomes. It was not. You see very distinct red versus green spots in these pictures. If it fused, they'd be yellow that you see in the lower left. So depletion of any of the 6 components of human HOPS complex inhibited fusion of dextran, Alexa Fluor 488 positive endosomes with the magic red positive lysosomes. So if the green dextran were transferred, there would be yellow staining like the controls at the left. So all 6 subunits are required for fusion, which you see on the right, but paralogs VIPR and BPS33B are not, as shown at the left. Now we'll look at iron transfer to the mitochondria. So lysosomal proteases require a low pH for their degradative enzymes to work. So lysosomes have to do a lot of work to maintain their low pH. There's that hydrogen pump again for H positive ions, the VATPase, which is on the right, constantly working. And in lysosomes, the inner membranes are highly glycosylated to prevent self-digestion. Those are the LAMPs, LAMP. And there's a big differential with other ions as well. Lysosomes must tightly regulate their ions. Hydrogen is 500 times what it is in the cytosol, and it's maintained at about 25 micromolar. On the side note, that VATPase hydrogen pump can be inactivated by bacillomycin A, which then tends to decrease lysosome enzymatic activity due to the pH increase. Something like that could be happening with the coronavirus. And calcium is 5,000 times higher than the cytosol. So let's look at how calcium is transferred to mitochondria through the lysosome efflux channel, TRPML1. This study is a great example of crosstalk between organelles. It shows how calcium is transferred from the lysosome to the mitochondria through 3 different mediating channels. Turns out that the mitochondria dysfunction of calcium regulation was actually a problem with the lysosome efflux channel, TRPML1. The lysosome couldn't transfer calcium through TRPML1 into the mitochondria. You see that on the right. They're studying the lysosomal storage disorder ML4, which is mucolipidosis type 4. These researchers learned that it takes these 3 molecules to transfer calcium from lysosomes to mitochondria, as shown on the left. This happens via TRPML1, which is transient receptor potential mucolipin 1, and VDAC1 in the middle, which is voltage-dependent anion channel 1 in the outer mitochondrial membrane, and the MCU on the bottom, which is the mitochondrial calcium uniporter in the inner mitochondrial membrane, leading to increased mitochondrial calcium in healthy cells. MFSA1 is an agonist that activates TRPML1, which you see on the left. So in healthy patients, TRPML1-mediated lysosomal calcium efflux leads to mitochondrial calcium influx. In ML4, TRPML1 is mutated and doesn't work, so calcium cannot leave the lysosome. It dysregulates the mitochondrial calcium dynamics, which you see on the right. So another note about TRPML1, it can increase the number of lysosomes. So during starvation, lysosomal calcium release through TRPML1 activates calcineurin, leading to TFEB dephosphorylation and nuclear translocation. So TFEB goes back into the nucleus to turn on autophagy and lysosome pathways. So let's take a look at their pictures of this contact tethering between the mitochondria and the lysosome. These are time-lapse pictures of HeLa cells. Lysosomes are the green circles because their membrane protein is labeled with GFP for LAMP1, and the mitochondria are red, tagged at the calcium center. The contact increases at 30 seconds after TRPML activation with MLSA1, which you see on the lower right with the yellow arrow pointing to increased mitochondrial calcium. Next, let's see the effect of calcium on neurons. In this experiment using MN9D neurons to study Parkinson's disease, they were trying to figure out how MPT works. It is neurotoxic. It causes cytosolic calcium to rise and overload and impairs autophagy and increases lysosomal pH, which decreases cathepsin B activity, which you can see in B. Treatment with calcium buffers like BAPTA AM in C restores normal cathepsin B activity as compared to A on the far left, and this ameliorates MPT. So you compare C with the control in A, showing that buffering calcium preserves the autophagy lysosome pathway and is neuroprotective. Next, we'll see that calcium is also important for normal repair of the plasma membrane by lysosomes. Another role of lysosome is to fix leaks in the plasma membrane, which tend to cause an influx of calcium. Detection of cytosolic calcium by the calcium sensor on the lysosomal membrane triggers the repair. Lysosomes are moved to the periphery by K1F5B bound to LAMP1 on the lysosomal membrane. They form a snare complex with the plasma membrane, leading to fusion, exocytosis, endocytosis, and repair. It's critical for lysosomes to move throughout the cell to do this kind of repair and delivery and pickup of garbage. So let's look at that next. Lysosomes move all over the cell. They move toward the nucleus in the retrograde when acid levels drop, and they move toward the periphery, that's anterograde, when acidity increases. This movement is critical for mitosis, for correct mitotic progression. The BORC complex is very important. If cells are genetically depleted of BORC-associated proteins ARL8B or PLEK-HM2, they show a significant increase in toroidal nuclei. Remember, they look like a donut. So in neurons, degradative lysosomes move to these distal axons. So if not, the axons tend to die. And this lysosome movement can be traced in microfluidic devices. This experiment is particularly cool because they were tracing the degradative enzymes from the soma into the axons. They grew the neurons in microfluidic devices to physically isolate axons from the soma and dendrites so they could see the axons clearly. They can label the enzymes in the soma chamber, then trace their influx into the distal axons, then test for lysosomal health via cathepsin L activity. They treated the neurons with baphyllomycin A, which inhibits the VATPase, the hydrogen pump, which stops the acidification of the lysosome, which you see on the right. If the degradative lysosomes aren't delivered to distal axons, the neurons tend to die. Here they show that cathepsin L is working along distal axons in the control on the left to maintain local degradation capacity, whereas treatment with baphyllomycin A on the right blocks cathepsin L activity. So lysosomal acidification is required for maintaining degradation capacity along these distal axons. They also selectively inhibited the activity of proteases, which we'll look at on the next slide. They wanted to selectively inhibit the activity of GKs, cathepsin B, and cathepsin V. So they used activity probes that showed they were specific. So E64D was used to inhibit caspase B to show that magic red is specific. And then they also use green BODIPI FL pepstatin A, which is an activity-based probe that detects cathepsin D. On the right, there is no signal from magic red, while the green signal for cathepsin D is strong. The signal density at the soma region was normalized and graphed. So it showed that, yes, these reagents were specific. And these cells had functional lysosomes. But what happens if the lysosome itself becomes damaged? I like this figure by Willow-Wen-Yu Yim from the University of Tokyo in Japan, published in February 2020. It answers my question, what happens to damaged lysosomes? They start leaking, and cytosolic proteins enter the lysosome, leading to heavy ubiquitination and emphasis of K63 link chains, triggering autophagy. And autophagy is a really interesting form of regulated cell death. The cell eats itself. It is a primary system for cleaning the body. This process delivers cytoplasmic or extracellular components to the lysosomes for disposal. It is a housekeeper using internal little balls of autophagosomes to fuse with the lysosomes for delivery. It removes or degrades unnecessary materials like misfolded or aggregated proteins, damaged organelles, cancerous materials, foreign pathogens, viruses, and microbes, and retains or maintains good materials like biochemicals, metabolites, and organelles that are required for survival, function, and development. And there's thought to be 3 types of autophagy: the macroautophagy, which is often just referred to as autophagy in general; microautophagy, which is when the intracellular lysosomes engulf small cytosolic substrates; and chaperone-mediated autophagy, or CMA. And these Hopf tethers are required for fusion, and inhibiting autophagy may have anti-cancer effects. Let's look at an experiment with that. So in our last example, we'll look at SSPH1. This is a novel anti-cancer saponin, and these Chinese researchers wanted to know why. And that's because lysosome function is impaired, which is seen on the lower left. SSPH1 inhibits autophagy by blocking autophagosome lysosome fusion, which induces reactive oxygen species, leading to caspase-mediated apoptosis, which is anti-proliferative. They use 3 inhibitors and graph them here. You can see that chloroquine acts on the lysosome to inhibit autophagy. NAS inhibits ROS on the right, which is seen in blue, and U0126 inhibits MEK, which is seen on the left in blue. I hope this data has stimulated some new ideas for your next experiments. ICT has many fast, easy, and reliable methods that can be used to evaluate lysosomal functionality, cathepsins, and cell viability in vitro, in vivo, and ex vivo. So which reagent is best for your experiment all depends on which instruments you are going to use for analysis and what other stains you're going to use it with. All of these kits are available online and through our distributors. Check them out and expand your research toolbox, including ICT's reagents. And that wraps up our presentation for today. If you have any questions, please feel free to send them to the chat box or email to help@immunochemistry.com, and we'll respond directly to you. Of course, you can always call us. We love hearing how you are using our products and helping set up your experiments. And again, thanks for tuning in today and for all the research you are doing. And thanks again to Allison Westcott, Nick Weninger, and Kristen Pauley, our marketing team, for managing these webinars. We here at ICT are proud to be part of the medical research community and provide these research reagents and kits that can help you understand lysosome biochemistry and cure disease. I'm Sally Head-Delquist, President of Immunochemistry Technologies, and it's been a pleasure helping you find a cure.